I'm going to say mulligan. Let's start from the beginning.
Okay.
Okay.
Hi, my name is Linnea.
I'm Bessia.
And I'm Linnea, and this is the Lemma Journal Club.
What is the Lemma Journal Club?
The Lemma Journal Club is the club where we read journal papers.
Boo.
I don't know what we decided on a tagline.
We decided that we were going to say the Lemma Journal Club is a show where we tagline here, read a paper, and today we are talking about the biophysical mechanism of mitochondrial purling. This is a pretty big paper. It's twinned with another paper that I might as well shout out at the very beginning. The other paper is Purling Drives Mitochondrial DNA Nucleoid Distribution. Both papers have a lot of the same authors. Gabriel Strum is the first author of this paper. There's Lefebvre on this paper, but it's Austin Lefebvre.
So it's a whole family of Lefebvres?
I do wonder, is it of relation, or is it? It's got to be, right?
It's got to be, right? I know that the father of Matt is at University of Minnesota. He does chlamydomonas research, so maybe it's a family of scientists.
Lefebvre, for our listeners, because it's a whole family of scientists, is spelled Lefebvre. So Lefebvre is how I hear people pronounce it. Nobody knows how the family pronounces it. It's also got, among the many last authors, Sophie Dumont, Suliana Manley, who's coming to give a talk here in a couple weeks, actually. And the last author of this paper is Wallace Marshall.
Well, so do we want to do a brief introduction to what the paper is? I think this one will go better than last time.
I just want to highlight, I think, while we're still talking about the authors, many of these authors are at least partially employed by Calico Life Sciences or Altos Labs, which are both companies, but that do, they're Silicon Valley aging companies. That kind of tints how I view this. I don't think that's entirely a negative thing, but I do think there should be that disclaimer that, there is a lot of aging money in this paper. Okay.
Well, anyways, we'll get into that.
Yeah.
I felt like they did a pretty good job of walking this line of saying it could have impacts on aging without actually making any claims.
I have okay.
Yeah.
I have issues, and I think that they're caused by the fact that Calico and Altos have a little bit of their ethics in here, and we'll get into that. You want to read the significance statement? So this was boring as shit the last time we did it.
Yes, but I think this one is more snappy.
Okay, so we'll try it. You read the significance statement. Let's see how it goes.
Okay. Significance statement. Mitochondria undergo dynamic shape changes critical for cell function. Beyond fission and fusion, this work identifies mitochondrial purling, a transient beads on a string morphology, as a fundamental mitochondrial dynamic observed occurring spontaneously across diverse cell types and physiological contexts, like T-cell activation, neuronal firing, and replicative senescence. We establish a unifying biophysical framework, the Rayleigh plateau instability, driven by forces of osmotic pressure, mechanical tension, and membrane elasticity. This study reveals mitochondrial purling as a key process reflecting the interplay between mitochondrial structure, membrane biophysics, and cellular activity, establishing its importance alongside fission and fusion for understanding cellular physiology, disease, and
aging. It does. I read that and I was like, man, this reads pretty similar to my job applications. I think a little bit of a convergent talking. So in addition to that, I guess we should say, normally at this point we say, why do you pick this paper? But this paper was essentially picked by both of us. This is a paper that's been up on the bioRxiv, again, with its twin, who I'll shout out again. Its twin is Purling Drives Mitochondrial DNA Nucleoid Distribution, have both been up on the archive for a while. This one came out in Molecular Biology of the Cell. Now, essentially, they're still uploading it. It looks like the resolution on the figures is pretty dog shit. Yeah. And we're on their GitHub that they've generously uploaded so that we can actually view their figures and their videos in decent resolution. It's a paper that's been talked about in multiple of our labs. It's a pretty wonderful exploration of something. I have a shocking amount of criticism, and I'm not sure how far to go into it.
Okay.
Given how wonderful it is. But so I just want to say it's a dual pick. we both picked it because it's very much in the zeitgeist right now. You wanted to add to that?
No, I think we've just been hearing a lot about this paper, and it overlaps with a lot of our interests.
And then I thought that they gave a really good description of the low-level physics, and I kind of wanted to read it out to catch everybody up.
Okay.
You think no?
No, I just have something else I want to talk about, but go ahead and do it.
Okay, so the paper is essentially about, you've got mitochondria, and mitochondria are essentially tubes, and they're made of bilayer membranes. This feels a little bit weird because I'm like describing it to like some imaginary audience rather than just discussing it with you like we normally would.
Yeah.
'Cause we know this whole game already, which is that a cylinder is not necessarily the minimal surface area shape for an elongated membrane tube. This is a quote from their paper. I think they spell it out quite nicely. If the surface is folded into sinusoidal curves, the same internal volume can be enclosed with a smaller surface area. Tubes tend to be cylindrical because the energy of bending a membrane is substantially larger than the energy of stretching it. So this is really important. This is Colm Keller's giant piece of wisdom that he put out on YouTube, right? Thin sheets are easy to bend, but hard to stretch. So what happens for a mitochondria is the mitochondria volume increases or decreases, but let's say increases. So now this membrane will need to stretch so that the surface area can enclose the larger mitochondria. It's really hard to stretch a membrane, right? You take like a mouse pad or a piece of paper or something, really hard to make it larger, but it's really easy to bend a thin sheet. Mouse pad, very bendable, paper, very bendable. So the mitochondria, instead of stretching out their membrane, they temporarily bend it. Thin sheets are easy to bend and hard to stretch. And so this results in this beautiful purling instability. This is the Rayleigh plateau instability. It's a well-known thing in physics. It's a well-known thing in bilayers in the soft matter community. Steve Granick has some terrific experiments showing this.
But yeah, the first reason we started talking about this paper was thinking about the Rayleigh plateau instability. And this is something that we learned about in fluid mechanics, right? This is a fluids concept. And so we started thinking about it when we saw in our own systems some things that looked like Rayleigh plateau, but were happening in membranes. And we had a big argument about whether we thought Rayleigh plateau could be happening in membranes. And it turns out that it has been solved thought about in the soft matter community, that since the membranes are liquid-like, where the components are able to move relative to each other, they can undergo this kind of instability.
Oh, my understanding, the thing that made me like at peace with this again was completely different from what you just said. Where I was originally like bilayers are not fluids. They have order. Right? And
yeah.
It was like, therefore, Rayleigh-Taylor does not apply.
But it's this block copolymer stuff.
It's this block copolymer stuff, but more broadly, Rayleigh-Taylor instabilities can apply to solids.
Okay.
That's okay. And so the trick is like, we generally don't think about surface tension and surface area things in solid systems because it's usually not important.
Yeah.
But solids have a surface tension term, and you can get Rayleigh-Taylor instabilities in solids. It's not a fluids only thing.
Okay, yeah, I should go back and read this Steve Granick paper.
I mean, the Steve Granick paper is doing it from kind of polymer physics, where we're kind of like in this murky area. But right, like the basic physics we have here is like there's a fight between surface tension and bending rigidity, right? And that's a perfectly valid fight to have in a solid just as much.
Yeah, I see what you're saying. Like, again, going back to Colm Keller's
you're probably not going to see a pizza undergo a purling instability, but it's not off the table.
Right. I think we should do a compliment sandwich style episode this time. I think
that's the way to go.
So maybe we start positive, we go into anything we have negative, and then we'll come back to the positive
Gabriel,
at the end.
Suliana has another paper. A lot of these people. Okay, so here I'll say this for everybody who's on the other paper is terrific, right? Okay.
That is not a compliment sandwich.
Okay, so then for Gabriel, right? Like, this is an amazing paper. This is a tremendous amount of work. It really is. It's super exciting. We're covering it for a reason. Everyone's super excited. The way that you've uploaded the data to the GitHub is super terrific. You know, this is...
Some of the images are like incredibly striking. Like, I love panel like figure 1B, right? The very first data shown in this paper is really beautiful, where you just see like these 5 pearls emerge and then disappear. You can totally see how this was a captivating motif to look at.
And for people who are, biophysics with a capital P for physics, who are coming from a physics background, I think the fundamental argument being made here is so clear and so obvious that it's almost trivial, right?
Yeah.
And so I actually think that a lot of the things about the paper is that they have to grapple with a biology community, which is like very interested in molecular mechanisms. And so there's a whole conversation going on here in this paper that I don't care about, right? Because I'm like, this is obviously true. Cool for pointing it out, right?
Yeah.
And I feel that about a lot of papers that are like this in biology, where it's like, oh, look, this little physics thing is happening. It's like, oh, yeah, cool. That's all you had to say. You didn't have to write a whole paper. But obviously that's not what's going on in the larger. For some reason, biologists have gotten into some sort of dogma that makes them unable to understand certain things unless you show it with 80 different inhibitors. I mean, essentially, right, like you just look at the geometry. Is it sinusoidal? Yes. Okay, it's a Rayleigh-Plateau instability, and that no amount of inhibitors or anything has anything to do with it. But that's not the conversation going on in this field, so whatever. Do we have other compliments to go before? I'll go in some light pokes, okay? Here's a light poke. I don't think it's a problem with the paper or anything. They use the snouty light sheet. Right? Great, cool. If you've got it, it's a lot of fun.
Yeah.
They do not. I mean, correct me if I'm wrong. I don't see any 3D images in this paper.
In the main figures.
In the main figures. Do they have them in the supplementary figures?
They have some supplementary movies that are 3D.
Ooh, they do. Do you remember? So they've got 38 supplementary movies. Here's another problem with this paper. Holy shit, guys. Synthesize just a little bit, right? Like, it really feels like this was rushed and that, it takes a long time to write a short paper. It takes a little time to write a long paper. 38 supplementary movies is too much.
I hear you. I mean, maybe they didn't all need to be supplementary movies for the paper, but I think it is a good resource for other people who are. I do, because if you're studying human fibroblasts, you'll be able to find the movie for human fibroblast mitochondrial purling, and you'll be able to find the movie for, your cell of interest mitochondrial purling.
Will you? Because you've claimed that there is a movie with 3D data.
So it might be 3D STED data is my only. So that would obviously not be on the light sheet.
So I don't see, but again, there's 40. So moving through these movies is very slow. Although they have put them up in GIFs, so that really helps. I don't really understand the justification for using the snouty light sheet. Right? You do lose a decent amount of XY resolution in order to have the ability to get isotropic volume resolution and to take volumes so quickly. And so if they were using the volumes, which I wish they were, because then they could do real curvature calculations and try to get some numbers here, that would be cool. But everything they present is in 2D. Why not just use like a resonance scanner and get the kick-ass XY resolution? That's my light poke.
Yeah, I mean, it might have been a poke from the reviewers. There's a sentence saying, based on these dynamics, a typical confocal microscope would miss up to 27% of purling events, depending on cell type. So they do
scanning confocal, but a resonant confocal microscope, like the Mako, will get 10 hertz images. Right? You only get a 2D slice, but they're only showing 2D slices. So what do I care?
Yeah. So I guess they think that you need the whole cell. And maybe they're just showing the 2D slices, but they did the analysis. I didn't go into their supplemental information on the analysis. But they do show some plots of curvature.
They show some plots of curvature. They never show a 3D reconstruction. They never show anything that makes me think that they're getting both the mean and the Gaussian curvature.
Yeah.
Which is annoying because like this is, we're talking about quantitative biology at its worst.
Yeah.
There's no measurements here. This is all qualitative imaging.
Yeah, I agree with this criticism. And the times where they did put in numbers. So at the end of this snouty paragraph, I have highlighted in red the last sentence, which says the shape of these pearls across cell types was highly conserved with an average width of 320 to 480 nanometers. That is so close to the diffraction limit. What are you talking about? That's at the diffraction limit. I don't know if they're deconvolving their images, but I would not trust those numbers personally.
And they're not like, those are not the important numbers for the equation at hand here, right? Like,
yeah.
I want measurements.
Well, peak to peak distance will tell you something about, right? That's what's the period of the sine wave?
I need my two curvatures. I need K1 and K2. You're correct. If they characterized that it was really a sinusoidal shape, that would be great for just saying, hey, this is Rayleigh plateau instability. But the other thing that I want is we can go and get measurements of bending rigidity, right? We can go and we can get measurements of surface tension. We can go and we can get measurements of K1 and K2 of the principal curvatures. In fact, they probably already have them. We can really plug this shit in, right? And you can compare it to the osmotic pressure, which you can calculate super easily, right? And you can see.
But that is not
physics work out.
Yeah.
And they're so close.
They're so close. Yeah.
And this is why I say this feels like a rushed paper to me. Is that there's a tremendous amount of information. There's been very little effort to curate it, right? They just essentially vomit it all up on you. And some people might think that's cool. I personally don't love it because it makes it hard to follow what's going on. And sometimes I feel like they're throwing out red herrings to make you not notice some flaws in some measurements. And I'll talk about the one that really bothered me. But it's so close to like real physics, right? They almost, and like, and like some of the figures are like bizarrely sloppy where there's just like giant white space in the figure.
Well, this is something that I think is a field specific problem. Or like, yeah, we were trained to never have white space in figures. But sometimes my current PI says, oh, just have the white space. Like, you don't want to be putting information that you don't need in a figure.
Sure, but just like some graphic design. I don't know. It like everything seems slapped together. They do the super resolution, and we only get to see one super resolution picture in the main figures. And really, like, I want a shitload of super resolution because that to me is the most convincing thing.
Yeah, so I mean, like, one thing that bothered me about this paper, for me personally, was they spent a lot of time being like, look, we see this in all these different kinds of cells. And look, we did all of the same experiment, and we over and over again in all these different types of cells. And why is this such a big part of the paper? You could have just said this in one sentence, and it's like, yeah, the first half of the paper, I felt. I
worry that this is something about the current zeitgeist in biology. To me, the thing is they're doing it all in just cells. I would like to see this in an organism. Like a singular cellular organism is fine. I want to see this in an organism. I'm not doubting.
Well, so I'm pretty excited we did this paper before the clammy paper that we still have on our list, because I wonder if we'll see anything like this in that data. Yeah. This is one of the things that I do love about the paper is it is a beautiful motif that you can now just look for in every mitochondrial image that you ever look at.
Bioenergetic motifs, that's my jam. Okay, so
how do we want to get into the meat of the paper? Because
what I want to do is I want to talk about their laser perturbations. Because it's a huge chunk of the paper, and I really don't understand what's going on.
Yeah, I agree.
Okay, so a major thing that they do in the paper is they essentially induce the mitochondrial purling with single mitochondrial position by applying a 0.3 milliwatt laser pulse for 200 milliseconds onto a mitochondria. And that this creates purling. Why? Why does this create purling? I really feel like this is one of two things where they're like, oh, look over there, and just like run away.
I just don't understand how this didn't come up during review. Because it's such, like, they don't even address what could be possible mechanism or what's going on there. They just observe that this happens. And, like, they use it as a tool. I get that. But yeah, the obvious question of why is this happening?
So then they use this as a tool to induce purling. And then they're like, what if we increase the osmotic pressure by using salts? That's, I guess, when you're changing PBS, you're really changing salts, is what they're doing.
Yes.
Yeah.
Yes.
They move salt up, they move salt down. They're like, oh, it's an osmotic pressure thing. We can probe it with the laser and see if we still get purling or if we still don't get purling. Right? The osmotic pressure is essentially causing the inside of the mitochondria to swell or to not swell. And so then the bending rigidity and the surface tension and the surface area all start to come into play. But why? What is the laser doing? This is because they do all these drugs and everything. The other flip side of this is that they use osmotic pressure a lot by changing salt concentration, which seems dangerous as shit to me. And I think they actually touch on it a little bit themselves. Come on, you guys know about PEG? Where's my PEG? Can you not put PEG in cells? You must be able to put PEG in cells.
You can get PEG into cells. Jing did it.
Polyethylene glycol.
Yeah.
Modify the osmotic pressure that way. Come on. Because, okay, so one of my concerns that I think is something that they might have actually missed is I think that it is possible, because this doesn't quite actually look like a Rayleigh plateau instability to me. It looks a little bit weird. It's possible that there is an electrostatic component to the purling, that the membrane feels itself, because it's a mitochondria. It's a very charged membrane at some points. The mitochondria feels itself and has a disinclination to come within the Debye screening length. So you're modifying the Debye screening length when you use these salts. I would like to, one of the reasons why I want the quantitative measurement so much is I want to see, do you need to start to put in electrostatic repulsions to justify when and how certain things occur? And this is my theory for the laser stuff. It's not there's two possibilities. One, you're poking micro holes in the membrane.
Yes, that was what I thought must be the mechanism.
The probable mechanism, which is not great.
Yep.
Second mechanism, very unlikely, but kind of cool if possible. You could be polarizing the membrane.
Yeah, so I do want to ask what polarizing is. So they talk about it a lot, and they're like, oh, the mitochondria get depolarized. And I have some vague understanding, but yeah, could you explain it for me and perhaps for anybody who else doesn't know?
So essentially, the mitochondria is a battery, right? And it's pumping protons back and forth along its inner membrane to try to create a proton gradient, to try to create a literal electric gradient of voltage, right? Lenny is now messing with her microphone, which is going to be a disaster.
I just want to sit up straight, but okay, well, I'll slouch.
You can lift the whole thing up. Better?
Yeah.
Okay. Getting back into it, the mitochondria is a little battery. It's got a literal voltage across its membranes. You can visualize that using a bunch of different tricks that we've talked about over the years and that I've used.
And so depolarization means when that battery is there's no longer a difference, voltage difference.
The amount of protons on one side is the same amount of protons on the other side. You're asking what the hell is with the protons in a biology talk, right? You take a hydrogen atom and you say, oh, you're in a liquid now, so you can kind of do whatever you want with your electron. And all that remains is a proton, right? Hydrogen is just a proton and electron. So we're just talking about protons getting shuffled around. So that's what the voltage is. And then you could imagine, right, you can charge molecules by hitting them with light.
Yeah.
Maybe a big pulse of light messes with the, battery a little bit. I doubt it, but it'd be cool. And I would explore it personally. But really, the first thing I would do, PEG. PEG and mechanical measurement.
So to dig down a little bit more into this PEG experiment. So like this would be the answer to the question we often ask of like, what is the one experiment you would want to see done? Yeah.
The one experiment I would like to see done, PEG. I think I could say that about almost any paper, but I'm going to really double down on this paper.
Okay, so I got a little turned around in this paper of, which direction of osmotic pressure is driving the instability, right? So it seemed like if the pressure is going up inside, then you get the instability.
Yeah.
So with a PEG experiment, are we increasing is there a way to increase the pressure inside?
Okay, so this is a good
I mean,
question.
maybe it's a different
So
size peg
it's
can give you different effects.
bending rigidity versus surface tension. As the mitochondria swells, the surface tension goes up, and the fact that the bending rigidity is high in a double membrane organelle becomes less important because that surface tension penalty is becoming larger.
Yeah.
So we either want to swell the mitochondria, or we can shrink them and see that the purling instability isn't happening. So if we swell them, we should get the purling instability. If we shrink them, we should suppress.
We should suppress, yeah.
So I think by throwing in PEG naively, using a big PEG that can't get into the mitochondrial
You should
membrane,
suppress.
you should suppress the purling instability, and it shouldn't be able to happen. That's what I want them to do.
Okay.
It's not as sexy.
But it's, yeah. So if you want to do the reverse.
So then for the reverse, you can do this thing that hurts me inside, which is you can use the ultra tiny PEG.
Yeah.
Which somehow PEG is polyethylene glycol. It's a little inert, carbon plastic, right? If it's small enough, normally it's a polymer. If you make it small enough, and Lenny and I learned this a couple of years ago, which blows my mind, it forms a liquid. It's just a plastic liquid. And it essentially, when you add it to water in that form, somebody described it this way. They said it makes water more watery, right? So I think you can go the other way with small PEG, though I'm not 100% sure.
Yeah. We could talk to, there's a great postdoc in Cliff's lab, Jing Zia, who does experiments with both small and large PEG in mammalian cells. So he might have some good intuition here.
What do we think? We've talked a lot about how we didn't like that they didn't do quantification, or we didn't like this. What do you think is intellectually beautiful about this paper, other than the framework? The intellectually beautiful thing about this paper is saying, hey, this is a Rayleigh-Taylor instability.
Yep.
And we're going to, just show that ad nauseum. But other than that, for the showing
I mean,
ad nauseum, what do you think was creative?
So I read this paper and I was like so excited. I spent the whole time texting Asta about all the things I'm excited about, because I think it's really summarized well in their final figure 8. These different possibilities for the biophysical possibilities for the arisal of these purling patterns.
Yeah.
And I found that really beautiful, and that they were able to, through these kind of battery of different tests, they were able to synthesize these 3 possible mechanisms. And I think that these are testable mechanisms within cells of is your cell responding, or is your membrane deformation happening because of pressure mediated internal or mechanism, or an elastic mediated or a tension mediated.
Yeah, so basically they say, look, there's 3 ways that you can really mess with the bending rigidity and the surface tension equilibrium of the mitochondria. The first is make the mitochondria bigger by a change in osmotic pressure, and the big thing that does this is salts. So you can imagine when there's a lot of calcium signaling or something going on, the mitochondria might be undergoing purling. That's the first thing that they say. I think that there's an electrostatic component of that. They don't mention it. The next thing that they say is, hey, you could actually mess with the double membrane. You could change the double membrane's properties. If the lipid rafts are behaving differently, you can reduce that surface tension term, and you can also change the bending rigidity term that way. And finally, they say, hey, mitochondria are pulled around by microtubules and kinesin, and maybe even actin and myosin. They can get literally pulled and elongated, and this is how we see a normal, classical Rayleigh-Taylor instability, and this can also happen here. Those are their 3 mechanisms. It's their main figure on the GitHub. It's their last figure in the paper, and it is a beautiful thing, because it's, okay, you want to talk about ad nauseum biological mechanisms, this little, cardiolipin thing does this little thing to the double membrane. Now you have a framework to do that. You're now talking about changing the membrane, and that will change this term.
Yeah, and so I found that really beautiful. And again, the complaint that I go back to is just, you really would have to dig deep into this data to understand what experiment you needed to do to test each of these different mechanisms, because they present so many inhibitions and salt changes and whatnot. And the connection between the raw data and the interpretation was lacking.
Is there something that you didn't understand? I guess we both said we talked about the laser. We talked about the mitochondrial potential. Is there anything else that we didn't understand? I mean, we've hit most of the things that we normally hit, and we're at about 30 minutes, so I think we could go. I'm going to do a little bit more shitting, and then we can go back and just say beautiful things about the paper.
Okay. Just one thing I wondered if you understood was this battery of inhibition stuff that they did to the mitochondria.
Yeah, they
page
did a couple,
9. Similarly, inhibiting electron transport change. A chain ETC complex 1, 3, and 5 with rotenone, antimycin A, or oligomycin, mitigated pearl curvature. So did you have an understand?
I do understand this.
Okay.
And I believe they kind of talk about this in the discussion. They talk about that the timescales of the purling are too quick to do classical seahorse oxygen consumption rate measurements to figure out how much ATP the mitochondria are producing. Because one question is, does this purling change the function of the mitochondria? I would really feel that it will not change the bioenergetics of the mitochondria, but they obviously feel that it really will. And we'll see how.
Well, I mean, the big question I think is what is the biological relevance here? It's such a beautiful a beautiful physics concept.
Purling drives mitochondrial DNA nucleoid distribution. Same people shuffled around. No, not entirely same people.
Well, we'll read it when it comes out. Follow up.
But so essentially, when you're doing seahorse, very commonly, you will use rotenone, antimycin A, and oligomycin, kind of one after the other. Oligomycin will jam up ATP synthase, right? This is the final bit of the electron transport chain that actually, this is the rotor that actually generates the ATP or phosphorylates the ATP, phosphorylates the ADP, right? Because you're putting the last phosphate on by using up this proton gradient. So
are they arguing?
If you jam up ATP synthase with oligomycin, right? Now you get a really strong battery because it can't release the proton gradient through its normal mechanism, right? And so all of these rotenone, so we're just kind of moving, they say this complex 1, 3, and 5, we're moving down the electron transport chain of what we're messing with.
Well, so my question is are those, is messing with those changing the membrane stiffness?
I think it must be, but
or is it changing? I guess you're asking.
That's not why they're using it. It's probably an offside effect. They're using it to mess with the potential, essentially, But are they? That's not why they're using it, right? They say the electron transport chain activity, but the main effect of messing with the electron transport chain, right, is to mess with that electric potential. And they talk a lot about how the purling occurs and then the potential plummets, right? And I think they've really gotten things mixed up here, right? It feels like throughout the entire paper they may have mixed up cause and effect over and over again, right? So one place they say to determine whether a specific ion or channel regulates mitochondrial osmotic pressure, right? But they're not measuring osmotic pressure. They're having difficulty measuring potential. It feels like there's a bit of a circularity of what is causing what.
Yeah, so I think I was just turned around enough in the paper that I wasn't always following, right? So they do show that calcium either goes up either before or after the purling. I could never figure out which direction that was going, and this again leads to my confusion about so what causes the pressure inside the mitochondria to go up. But anyways, the question...
This is my problem with the paper.
Okay.
Is that this is a flood the zone paper.
Yeah.
You've got 40 supplemental movies, right? You've got figures that are beautiful, but often unreadable. This is, I think, the least I've ever looked at graphs is figure 1 graphs. Who the fuck knows what they're trying to show? They're making every statement under the sun, and then the next paragraph, even 3 sentences later, will be a non sequitur where they're talking about an entirely different thing, right? Right after this relating ion channels to osmotic pressure, they are right off the gates and they're showing, oh, this revealed that mitochondrial tubules enter a flaccid shape, identical to hyperosmotic shock. And it's like, and here's, supplementary appendix, movie 17, figure supplemental 3G. It's a huge tract they go over. It's like this massive land grab for some reason. A lot of it comes down to the point where it doesn't feel like science anymore. It's just like pop sci. You don't just say every single thing you think, right? You don't just yap and yap. You gotta really show a thing, show that one thing, show it really well. And so you're showing the mitochondrial purling, you're showing it really well, focus down on the biophysics of that, appease the biologist diehard shits that need you to use 18,000 inhibitors to show something obvious. But then, they're all, yeah. Okay, I'll say one more statement. I keep saying that. I just see the next sentence right after that. It's because of this flood the zone paper that, like, every other sentence I have highlighted, they use PFA to try to view the mitochondria, right? This fixative. PFA messes mitochondrial shape. Everybody knows that. So they dope with glutaraldehyde, which is what everybody does. Glutaraldehyde is a stronger fixative, right? It does 2 formaldehyde bonds instead of 1. Or I didn't say that quite right, but whatever. And they get a better fixation of the purling. Don't explain why. Zoom on. And do they even show a picture of a fixed mitochondria? Because if they can fix the mitochondria in the purling state, then I want really fucking beautiful geometric analysis. Also, they say it's not that it's cell-dependent for glutaraldehyde. I bet you it's glutaraldehyde dependent and it's not cell-dependent. Glutaraldehyde, vial to vial, stupid amount of variability, and it ages super quick even in the fridge. I found glutaraldehyde very obnoxious to work with.
Well, for this figure, for this 2C, I think, so PFA is the last panel. I was a little confused about whether these were reversible processes. So I thought that it was all reversible, but then they put on PFA. And how long do you need to leave PFA for it to be reversible?
What makes you think that they're reversible? Because normally when I think about adding a drug, I don't really think about, like, you can try to wash out the drug, but you don't really know.
Well, but like digitonin is not a drug. It's just a chemical.
Okay. And you think that they're trying to wash them out? Why do you think that?
I thought they said something about washing it out, but maybe I'm, again.
I mean, definitely they ain't washing out the PFA.
Okay.
I mean, you could wash out the PFA, but it's too late.
So in the shitty version of the figures, I don't know if you only looked at the bioarchive version.
I'm now only looking at the GitHub, but yeah, sure. In the shitty version of the figures.
Okay. So there's
and just for our listeners, like this is
MBOC's fault, I think.
This is molecular biology cell fault. I've never even like the letter A or B on the caption is barely legible. So it's like 6 pixels to the A.
They changed figure 4.
Okay. Cool.
And they do include some FibSem data of this purling that they didn't take, yeah. Right? But you did say, like, you wanted to see these beautiful pearls.
This ain't
it. This ain't it.
This is.
I mean, it's somebody else's data, but sure.
Maybe it is if you increase the resolution to more than 20 pixels.
Yeah. I mean, I was pretty disappointed to not see this. And it also isn't on the GitHub, which, maybe they just have to wait till it's out.
Yeah. So generally when you're publishing a paper, for people who don't know, you're not allowed to make changes to like your public GitHub bioarchive version until the paper has come out. And even then, it's a little bit shifty. So they are kind of locked in on what they're allowed to show at this point. This new figure 4 sucks. What is this giant white space in the top right? It looks like they've just like deleted accidentally a figure panel.
Yeah, probably they deleted a figure panel on purpose and didn't.
Wallace Marshall, on the incredibly unlikely chance that you are listening, first of all, I am extremely sorry and please don't hate us. But also, hire a graphic designer. I think you're rich enough that you can do it. Hire a graphic designer. I mean, certainly Calico and Altos.
Okay, yeah. So now that you bring them up again, what is your concern with them?
I think that the reason why it's such a land grab and why it's so sloppy and why it's such a like flood the zone paper is because that this is in some sense not an academic mentality. This is how you write a paper if you're a business, right? This is do the most science in the least time, break things, move fast paper. And there's something kind of good about that, but I actually think this paper has gone too far. It's too fast, too many broken things. There will be things that are wrong in this paper that are going to be a bitch for future PhD students to fix. Like, people are going to dedicate 5 years of their lives to fixing something that these people did for 10 minutes. And that's me saying that. I feel like I'm pretty work fast and break things. And I think when you've gone too far for me, you've really gone too far. Like, I think the old guard like Von Meer would really be furious at this paper. I think he'd walk out.
Yeah, I hear you. I definitely think that it's like there's parts of me that agree. The part of me that doesn't agree is just I hadn't thought about this phenomenon before this paper started coming up, and the power of thinking about membranes in this way. That's what I think should have happened, is a smaller paper where they just show. I mean, I think just use the same data and tighten everything up, right?
But this is why it feels like a company decision to me. It's like, if you're in science, you spend a few years working on a project, you spend a lot of time deciding what data not to show. You say, oh, we've seen this, we've seen this, we've seen this, we've seen a big zoo of things, but you don't show the whole zoo of things.
Yeah.
You focus down onto the main point so that people can really get the main point and that other people can focus on future points. And here, they show the whole zoo, which is much faster, much more efficient, much more impressive, but ultimately makes the field much harder to make.
Yeah, but if this paper, I mean, so I understand long term that this, I mean, now we're getting into philosophy of science things, but that long term this paper might be destructive. But if it's so timely for me, like for putting together my faculty proposal and for thinking about projects that are going on in the lab currently, that if it had taken an extra year to come out, that would have impacted my ability to do science. And so go fast has its benefits.
But just publish a paper half as long. And also, if you're going to use the Rayleigh-Taylor instability, write down the Rayleigh-Taylor instability.
Yeah, that's a good point. But it's molecular biology of the cell. You can't put any equations in.
No, come on. Che Hong has a paper in molecular biology of the cell that is such advanced quantum mechanics that, like, I talked to him about that paper for 2 years and I still can't understand it.
Okay,
right?
Yeah.
And he got like paper of the year in molecular biology of cell for that paper. So they're not math shy, and these people on this paper are not math shy.
Yeah.
So give me my equation. The biologists shouldn't be so scared of math. They can do it. They like math too.
Okay, do we want to sandwich it?
Yeah, we're going to sandwich it with the following question.
Okay.
What do we think are the deepest takeaways from this paper? What did we technically learn? How does that move us forward overall?
The deepest takeaway?
Yeah. So I think I'll give my answer. They say this thing at the end about how does this Perling instability modify the function of the mitochondria? Specifically, how does it change the oxidative phosphorylation, the ADP phosphorylation into ATP? How is it changing the energy production of the mitochondria? And it seems so obvious to me that it doesn't, that I actually didn't consider this question at any point. And I actually appreciate them putting it in the discussion, because then I have to say, okay, wait a minute. It probably is going to at least have some epsilon change. There is, a momentary change in potential, even though this happens very fast. And that's certainly going to change the energy production. And they bring up some good points, right? Like, this is happening a lot in some cells. In cells where it's happening a lot, you're losing your potential a lot. And so actually, this probably will change the energy production of mitochondria. And there's a reason for them to not do this Perling instability if they really do have to lose their potential in order to undergo the Perling instability. They say, we hypothesize that energy production will follow a biphasic effect, with increased oxidative phosphorylation as the IMM, which is internal mitochondrial membrane, I think, but I don't see them actually say that anywhere, but I'm sure they say it earlier up in the paper. As the internal mitochondrial membrane increases in tension, followed by a precipitous decline in oxidative phosphorylation upon crossing the Perling instability. It's cool. I don't know how the hell anyone's going to measure that. I think you've got to measure it with a fluorophore. You've got to go get, put a fret pair on the ATP synthase, or my ATP dye might show this. Again, if you guys are listening, ATP Red 1, see if you can catch this. But I think probably not. I think it's too fast. But fluorophore is the only way I think you're going to possibly catch it. And that's been interesting to me because I don't think that. I know your whole shtick is you think about form and function of organelles heavily on the function of organelles and attaching form to function of organelles. I don't. When people talk to me about my mitochondria data and they say, oh, is it fragmented? Is it continuous? I say, I don't care because that's not how I'm thinking about these problems. And so I really appreciate that they put in the discussion, they forced me to think about it, and now I have to think a little bit about it.
Yeah, and to this question as well, I think the difference between and they talk about this in discussion as well at the very end of the connectivity of the microtubules, not microtubules, mitochondria. I know.
They say mitochondria tubules, and I feel like I'm having a stroke.
Yeah. But for the connectivity, right? I had always been like, who really cares if your mitochondria divide? Like if you look at images of mitochondria and cells, they're just all over the place. They're in like vastly different shapes. They're not these nice little oblong things that you see in your textbook. But here is a mechanism by which, like, if you mess up the osmotic pressure in one tiny area and you're a totally connected mitochondrial network, you're messing up the pressure across the whole network.
Here's what I don't understand. They show this over 100 microns in one case. Right?
Yeah.
Unbelievably large cell. Cannot imagine. They laser poke the mitochondria in one place, 100 microns elsewhere, it's still purling. What?
Yeah.
I mean, normally, if you try to do that like in a finite element simulation, right? Normally, you're just gonna have some losses as you move in space, right? And those tensions and those forces are gonna relax, and you're not gonna get purling 100 microns away. I mean, I guess you do.
Well, maybe you're very close, like you're very close. The whole membrane is very close to the purling instability.
And you just can't.
And you mess up the region close by a ton and the region far away just
that makes me start
enough.
To think about all sorts of diffusion in a cell. I'm starting to think about it as a little bit of a slow thing. I used to think about it as 500 microns squared per second. Now I'm thinking about it as more, it's like 10 microns squared per second. It starts to make me think about you could use mechanical signaling in this manner to span incredible distances almost instantly.
Yeah, so I do wonder, like, did they talk about the time scale for that purling to happen? I don't think so.
Maybe about it a little bit.
Okay.
They're like, we have to be so fast in order to see it.
No, but I mean the time scale for when they poke to see it 100 microns away.
They don't.
Yeah,
and there's information there.
Yeah. So I would love to see that. Maybe it's in their supplementary movie, 40 supplementary movies.
Anyways, it's awesome. I think we'll see
really provocative
written about this in the next 3 or 4 years. Certainly, if I could drop everything, I would absolutely love to play with some of this. I've got so many questions. I got so many things I think I could do, but it will have to be people who are moving into new PhDs, new postdocs, who are in positions who can follow up on this.
Oh, and the other thing I loved about this paper, just for a final cherry, is that so much of it is done with dyes. So you don't have to do genetic perturbation, which means it can be done in non-model systems. So they use tissue culture for most of this paper. This could be done in any organism.
I think they're using dyes for the record just because they're trying to go fast.
Yeah, I understand that. But I think it makes it really...
but they're doing things.
You can check this in a lot of systems.
Could be putting in dyes. I mean, they could be putting in genetic.
They could be putting in genetic things, yeah, and that
I'm surprised they weren't forced to by the reviewers. Good on them. Good on the reviewers for not schlepping them out to that.
Yeah, I don't think it would change. I mean, I personally don't think it would change the results, but I am excited because it makes it easy to reproduce in like living organisms.
Yeah, let's go see it in Chlamydomonas.
Yeah, I mean, maybe next week we'll see it in the paper. Maybe it's there.
I've got no more notes. Just my biggest note, all capital letters, is what does the laser do?
Yeah. I feel like it's poking holes in the membrane and causing osmotic changes due to that, but...
causing potential difference. I kind of feel that, again, if there's an electrostatic term added into the Rayleigh plateau instability, just messing with the potential...
Well, so this goes back to my question that we didn't really answer of what all of these inhibitions, mitochondrial inhibitions, are doing. Is if they could be changing the stiffness of the membrane, or they could be changing the electrostatics of the membrane.
But you can do that orthogonally, right? Like, well, not quite, but you can go change the stiffness without changing the electrostatics.
Sure, but they don't compare those. They just say that they do these inhibitions and it decreases the rate of
But the way to do this
Pearl
is
curvature.
not to sit there with 80,000 inhibitors and a bunch of really sloppy measurements. The way to do this is to go measure the physical measurements, which this is a rare system where I think you can actually get all the bits of physics.
Yeah.
And to see, do I need an electrostatic term to explain everything? Is everything purely explained?
Just a curiosity question. Have you read the other paper?
I've perused the other paper.
Okay.
Have they done this?
Yeah, that's my question.
I think that they haven't from my perusing of it.
Okay. Maybe
Actually,
it's...
I might have actually read this when Asta first pointed it out to me. Yeah, and just scrolling through it again, I see that they don't. I always thought of the other paper, Perling Drives Mitochondrial DNA Nuclear Distribution, as the biology version of the paper.
Yeah.
And this paper as the physics version of the paper. And now after having read this, I'm like, well, this isn't really physics. There's no equations. There's no measurements. This is very qualitative. Again, we love the paper. The other paper is just they're both biology papers.
They're both biology papers, yeah.
No real physics paper. I think that this is going to be a theme that we see a lot, is you take a word from physics and you bring it into biology, and that's biophysics, right?
Yeah.
And it's going to take... And then people start to crank the wheel of doing the actual physics, right? But it's going to take a bunch of people getting much more comfortable seeing math and much more comfortable moving away from their dogma of this protein leads to this protein leads to this protein, or RNA goes to RNA is how people tend to think about it now. And yeah, before we get to see the math come in on the very first paper where it belongs, I think it'll be a while. But this is okay. I like this. This is all you have to say is the Rayleigh-Taylor instability and show me a picture, and that's it. Could have been one figure. It could have been like a math paper, right?
It could have been one figure,
word, one picture, no explanation. You don't even have to say it's a mitochondria. Yes, and that could be the paper, right?
Yeah. That's really what I took away from this paper, and I'm really grateful for that. So great. Okay.
Yeah, and so thanks for listening. We'll be
uploading on Mondays.
We're uploading on Mondays. Tell people about this podcast. We are trying to reach, I don't know, some audience, and probably the only people who listen to this right now are parents. So parents, tell your friends. If you're a scientist who we're talking about and we've made you sad, we think that your papers are better than our papers, right? So, it's a hard... love what you do, and you'll take everything super personally all the time.
And have no boundaries whatsoever.
Yeah. We're trying to be as positive as we can, right? There's something like inherently negative about Journal Club, and we're trying to be positive, and we have questions to make that happen. We're going to try some other formats in addition. These are not formats to replace this format, but we're going to try to have some additional formats that we occasionally do not on a schedule, and we'll probably be uploading one of those this week. So look forward to that. And thanks for listening. Give us 5 stars. Bye.
Bye.